I'll tell them right now. The very first that came to our mind is, well, if you have so many genes oscillating with this harmonic, beautiful oscillation of gene expression, we get to accept the fact that there must be a system at the chromatin level as at the epigenome that needs to allow this breeding of the whole system so that we have some type of a chromatin remodeling that is also circadian. On the top of it, if there are so many genes that are regulated, that means not all those genes can have to do only with circadian motorial activity, but they must hit additional important functions in our body. And I'm not going to go and tell you, but of course, when you go and look at all the lists of those genes regulated by the clock, yeah, you have things such as metabolic enzymes and critical elements of our Krebs cycle, and cell cycle, and so on. Now, another way to look at this is that looking at mutants that people have generated in the mouse. When you go look at mutants of the clock gene, these mutant mice not only have a problem in circadian behavior, but also have a defective fat metabolism. They're basically obese. Per one null mice have display higher addiction to drugs of abuse. These are cryptochromes, the repressors of the clock system, have drastically impaired liver regeneration. And so there are problems in the cell cycle. And per two null mice are prone to a number of cancers, specifically lymphoma. So that tells you how broad is the implications of the control by the clock system into our physiology and pathophysiology. So anyways, basically, conceptually, we had to invoke the possibility that a global mechanism of gene control involving chromatin remodeling had to work in order to allow these many genes to oscillate. And I'm not going to go through this in too much detail, but the very first implication, the very first information came from work done at least 10 years ago. By looking at that system I showed you before, that is just shining light to our eye, actually in this case it was a mouse, in the middle of the night, and there by just 5 minutes, it's called a light pulse of 5 minutes, so just normal white light. And you induce very physiologically the retinal hypothalamic tract, and you induce those neurons in the suprachiasmatic nucleus. When we go and look at what happens to those neurons, in using a phosphoserine 10 antibody here, the H3 tail, you see a very beautiful phosphorylation of H3 in response to a single light pulse in the middle of the night. And only in those neurons, nowhere else in the brain, and specifically during nighttime and not during daytime. So that meant there must be some type of kinase that is involved in the phosphorylation serine 10 response to light in those neurons. And we're studying that kinase. I'm not going to go to that today, but what I'm going to do is to introduce you the other question, which is how to somehow establish the circadian rhythmicity at the level of the chromatin into a more stable type of signal. And one signal that is a bit more stable than phosphorylation is acetylation. We can talk about methylation during questions if you want. But this leads us to look at our special K, the K14, which has been already introduced at this meeting. In a study that our friend here, Peter Chung, who was the chair of the previous session, in a study we'd done several years ago, now almost 10 years ago, together some studies by Shelley Berger, we're showing that serine 10 phosphorylation here actually induces a much better acetylation at this K14, and sometimes establishing a better, let's say, basis for activation of gene expression. So the question we had in the lab, and I'm going to go through this a bit quickly because it's been published, is what is the circadian hat? So the hat that will be responsible for the acetylation of K14 for all those 15% of the genome, or let's say call it the circadian epigenome. When we use an antibody that will recognize specifically that acetyl-14, and we'll look at the acetylation, this is now a clock-responsive gene that is present in liver or in the brain, in some parts of the brain. You can see that that acetylation K14 is nicely circadian. And this is true for a number of the genes we want to look. So we can imagine that there must be a system, must be a hat, that is responsible for this circadian modulation of acetylation. And to make a long story short, we found that that transcription factor involved in that autoregulatory loop called clock, that people thought for long 10 years to be a simple transcription factor, is in reality a hat. And it's a very peculiar hat. I'm not going to have the time to tell you about it now, but it's an interesting molecule for many reasons. All right, so at that time, we had questions which were the following. And we have now a new type of hat, possibly controlling the circadian gene expression in acetylating H3 tail, at least in K14. The other questions were, okay, what about possible acetylation of known histone proteins? And the other question was, if we can call clock as a writer of acetylation, what could be the eraser? What will be the HDAC to remove that acetyl? And I'll answer both questions very quickly now. So the first question was addressed, and is important because we all know the hats not only acetylate histones, but also acetylate a number of critical proteins in the cell. A couple examples shown here. We all know that, let's say, acetylation of p53 is critical to its, and it's the acetylation to its tumor suppressor function. So finding important targets, known histone targets for clock, was critical to understand how much the implication clock will have into acetylating a number of proteins in a circadian manner. The answer was that at least we found one element. I'll tell you more about it in a second, but that is acetylated, and that's its own partner. Remember those two guys, clock and BMAL. There's a dimer of transcription factors. Clock is a hat, and clock is acetylating its own partner, BMAL, at one specific acetyl. One specific lysine here is a K537. I'd like you to check that there's a K538, also nicely conserved, which is not acetylated, and that's why I like biology so much. Now, we made an antibody against that specific K537, and that antibody showed that we can go and detect highly specifically acetylation of this BMAL substrate for clock. And nicely, using that antibody, we can see that acetylation also is circadian. This is now fibroblasts, which are serum shocked. By serum shock fibroblasts, you can induce the circadian clock in those fibroblasts, and that acetylation is also nicely circadian. I'm not going to go into the details, but what is really important to say is that if we mutate this K537, we block totally circadian responses in the cells, in the animal. So this is really a critical switch. A couple of things to say about that particular site, and I think it's really cute to see that K537 in BMAL is preceding the BGGXS serine that is highly reminiscent of serine 10 in histone H3. Remember that serine 10 is phosphorylated in those neurons in the suprachiasmatic nucleus in response to light. The same sequence is present in both substrates for clock. So we can imagine to go look for this type of domain and this type of motif in a number of proteins. That's what we're doing in the lab, and I'm going to go again into the details. But questions could be, can clock go and acetylate known histone proteins that are involved in the clock machinery besides BMAL? A good question. What about metabolism? What about nuclear receptors, which are so crucially important for our metabolism and circadian physiology? We tested something like 12 nuclear receptors, and I can tell you that only one has that motif I showed you before, and that motif is very nicely acetylated, and we see that that particular nuclear receptor is critical for metabolism and inflammation. What about the cell cycle? Yes, we can go look F-box proteins. We can go look at P53. We're doing that. Another really interesting question is about sleep. Of course, we all know that our sleep-wake cycle is somehow related to our circadian cycle. And so the question is like, what about regulatory peptides for sleep? What's the relationship with the clock? Could be an enzymatic relationship through acetylation. And that's what we're doing. Now, the other question was, I hope you remember, if clock is a writer, so clock is a hat, which one is the HDAC? And that is work by Yasu Nakahata, an exceptional postdoc from Japan in the lab. I'm not going to go through, again, all the introduction about HDACs. This audience doesn't need it, but we all know there are several classes. Our attention, for a number of reasons, was attracted by one class, the class 3 of HDACs. And I'm sure most of you know that the class 3 includes a